In this module, we will again talk about the oxygen requirement in any fermentation process, especially on a large-scale fermentation. So as concerned our previous discussion, that as much as the carbon will be utilized, the oxygen demand will be high. And if the cell mole will be produced, and then there will be 3 times more oxygen is required as the amount of the cells have to produced. So it must be remembered that the high carbon content of the medium require high oxygen content in the medium. So the gram biomass produced by gram consumed of the carbon source require more oxygen. So if we say that the need of the oxygen for this purpose, so it is inadequate to the base provision of the oxygen for the fermentation, because number of factors involved on the supply of the oxygen. So if we want to estimate the overall demand of the fermentation process, it is very difficult to calculate. But because during the fermentation process the oxygen demand remain increasing, we can say that varying with the passage of the time, fermentation time. So the effect of dissolved oxygen, because the organism has to utilize the oxygen just in the form of dissolved oxygen. So the effect of dissolved oxygen concentration on a specific oxygen uptake rate, which you can see here, the QO2, that is the mole of the oxygen consumed per gram of the dry weight of the cells per hour, has been, we can see in our next slide, the relationship of this specific oxygen uptake. So it must be seen by the specific oxygen uptake rate increase with the increase in the dissolved oxygen concentration up to a certain limit, we refer to as a critical stage. So you can see this graph on the x-axis, it is a dissolved oxygen concentration, and on y-axis you can see here that is a QO2. So as well as the rate of the oxygen is increasing, so you can see here that if the concentration of DO2, dissolved oxygen concentration in the medium, and the number of the cells are constant, we can see that their utilization and uptake of the oxygen gradually increase with the increase in the dissolved oxygen. But that will be constant on a certain point. On that point, that is known as the critical point, because at that time each and every cells has the specific amount of oxygen that he needed. Then we can see that that is the critical need of the fermentation process at that time in a specific concentration of the cells. So you can see in this slide that the critical dissolved oxygen concentration for the range of microorganisms. In this table there are the 4 different microorganisms: Azotobacter, Escherichia coli, Saccharomyces, Penicillium chrysogenum, at different temperature. As we know that the solubility of the oxygen and the requirement of the organism vary with and the oxygen effect vary with respect to the temperature. So 0.018, 0.008, 0.004 and 0.022, these are the critical dissolved concentration of the oxygen in millimoles per liter of the medium. So in next slide you can see that the oxygen requirement of the different organisms with respect to different substrate. You can see in this table that there are the 3 different kind of substrate: glucose, methanol and octane, and the oxygen requirement when the substrate is glucose by Escherichia coli is 0.4 gram of oxygen for 1 gram of the dry weight, and methanol is 1.2 and octane with Pseudomonas 1.7. In the start of this module, I told you that the oxygen requirement with the carbon type increased. So you can see the carbon compound and carbon items in octane is more than that. So that's why the oxygen demand is more in case of the octane as respect to methanol and glucose. So the different carbons have the different oxygen requirement for fermentation processes. So the maximum biomass production can be achieved during the fermentation process if we can satisfy the oxygen demand of the cells. So in the later module, we will talk about how we can calculate the oxygen demand for a particular organisms.